5 million people are losing a war against their own DNA, and we call it lupus. For decades, the gold standard of therapies have been toxic steroids which ravage the body as much as the disease itself. These patients are trapped in a constant cycle of systemic deterioration because our best medicines aren't precise enough. Why aren't they precise? Because we're assessing drugs with outdated metrics. The race for T-cell engagers, known as BiTEs, assesses affinity, a singular point of binding strength in a static space. However, in the vasculature, cells experience multiple points of binding and dynamic flow. A BiTE can have perfect affinity on paper, but detach in the body due to flow. And when this BiTE detaches, the therapy fails. This is known as the affinity gap. We aim to close said gap with a microfluidic chip that mimics the hurricane of the human microvasculature to assess the new metric, cellular avidity. Our team has already shown that cellular avidity is highly predictive of in vivo efficacy. We aim to use this platform to develop the next generation of superior biologics to treat lupus. We have secured £1 million in grants and are seeking an additional £3.8 million to complete preclinical testing. At Evidence, we are building the future of immunotherapy, one stable bond at a time. Thank you.
Hey, thanks. Is this a platform company that you're building, or do you hope to use this as a way of maybe identifying novel therapeutics?
Yes, so it's kind of a mix of both, where we have this platform, and if our company's aim is that if we were to have candidates for, say, for lupus, we test it and put them in our device and test them for avidity, and then from then on, we'd be able to put them through to clinical trials.
And a follow-up to that, actually, so how many candidates would there be in the market at the moment, and how many clinical trials are there for lupus at the moment, to get a sense of your market size?
So I'm specifically working with bispecific T-cell engagers. Currently, there are no FDA-approved biologics for lupus. We are looking towards a CD3 and CD19 bispecific T-cell engager. However, well, there is one. However, it's not approved for lupus, so it's kind of contained within a completely different space. We also are looking, another kind of competitor would kind of be what's kind of trending right now, which is CAR T-cells. There are companies such as Kyverna and I believe Cartesian Therapeutics, which are looking to use these to also treat lupus as well.
So avidity and affinity is important for monoclonal antibodies. Why did you choose lupus as the disease to go into rather than another disease where a monoclonal antibody would be useful?
I think, once again, it's all tying back to the idea of using the development of bispecific therapeutics is what our research project was kind of focused on. There's great concentration with bispecific therapeutics towards cancer, but not much within the autoimmune disease. So we also could look into other autoimmune diseases, but specifically for lupus, also we're thinking about targeting America. They have the second largest incidence or burden globally, which is about 302,000 patients, and of those, with our biologics are typically used to treat those with moderate to severe lupus. I think that represents a large financial opportunity, as we see with competitors such as Safnello and Benlysta, which are currently leading the market. They've brought in about quarterly revenues of about $420 million. So we'd expect to have that same kind of gravitas when we enter the market.
Thank you, TJ.